thank you, John, for that introduction. I hope to share some successes. John has been overly kind in stating my accomplishments, I should say, but hopefully what I'll be able to present to you today will convince you that we are on the right path. So I am an immunologist by training, and my lab is essentially interested in the mechanisms of innate immune defense and how these mechanisms perhaps impinge on development of autoimmune diseases and cancers, which we understand are very complex diseases, many times associated with dysregulation of the host immune system. And with regard to any kind of thematic relevance to the symposium, I think, in part, the future of health actually lies in understanding mechanisms of disease and perhaps trying to gain enough insight to, at some point, intervene with these mechanisms. So as I said, my lab is interested in the mechanisms of innate immunity, and we now understand that the innate immune system is our first line of defense against pathogens. Bacteria, viruses, fungi, parasites, even stimuli or stress signals released from our own dying cells can activate the innate immune system. So I thought that I'd start this presentation by briefly taking you through the initial events that occur following infection with any given pathogen, and in the process, introduce you to some of the key aspects of our work, and then go on to describe two stories, one of which is published, one of which is unpublished, to give you some idea about how we think the immune system can regulate this very important balance between host defense and the tipping point into disease. So we know that bacteria such as Salmonella can invade the epithelial barrier, and when they do so, they're typically taken up by macrophages, which underlie this epithelial barrier, and are sensed by many different classes of what are called innate immune sensors. At least one such class of receptors is the NOD-like receptors, and this is the largest family of innate sensors. The NOD-like receptors, or NLRs, as I'll continue to abbreviate them, are essentially a family of sensors, and these can recognize both pathogen-derived signals as well as I previously mentioned, danger signals released from dying cells. They typically respond by releasing a set of cytokines. The most common ones are IL-1 beta, interleukin-1 beta, and interleukin-18. These cytokines can induce neutrophil recruitment, particularly IL-1 beta, and this constitutes what we think is the innate arm of the immune response, while IL-18 can act back upon T cells to produce interferon gamma, which is a key adaptive cytokine. And so in this whole scheme of things, in the past and present, I've been interested in essentially understanding the interaction of Salmonella with intestinal epithelial cells, its interaction with macrophages, that is how pathogens start to establish niches within host cells, like macrophages, upon systemic dissemination. And Ajay in my lab is working on this. You might perhaps have seen his poster yesterday. We're also interested in understanding the signals that activate these innate immune sensors. Like I said, these can vary from being pathogen-associated signals to danger signals released from dying cells to stress signals. And in doing this, we typically take population-level approaches. We make cellular extracts, Western blot for different components. And at some point, it became clear to us that in order to really understand the spatial aspects of innate immune activation, we needed to start visualizing the components to see how and where they come together. And so using imaging, I observed during my postdoctoral work that at least one of the NLRs, that's NLRP3, is activated in association with mitochondria, which are essentially subcellular structures within the cell. Collaboratively also found that cytokines like IL-1 beta and IL-18 can regulate innate defense in lymph nodes, particularly T cell responses. But the larger goal of my lab here is really to undertake a systems biology approach to understand the functions of many different members of the NLR family, of which NLRP3 is essentially only one member. So at this point, I should reiterate again that the immune system is, of course, involved in host defense against pathogens, but it is a double-edged sword. If you have underactivation of the immune system or overactivation of the immune system, it can lead to immunopathologies. And NLRP3 in particular is one such protein, which we found associates is activated in association with mitochondria. And so I'll briefly describe that data, which is already published, and tell you where we are in gaining insights on that front. And so why is NLRP3 so important? It is required for detecting pathogens, but on the other hand, gain-of-function mutations in NLRP3 are also associated with severe human autoimmune diseases. And beyond that, NLRP3 is also involved in the inflammatory response to dying cells in the absence of any infection. This is essentially what we call sterile inflammation, where there is no detectable pathogen within the system. And it's been implicated in sterile inflammation in these many different and diverse types of autoimmune and autoinflammatory diseases. And so while there's little doubt about the role of NLRP3 in human disease, we still don't quite understand the mechanisms of its activation. And when I started this work, the dominant paradigm in the field was that NLRP3 is present in an auto-repressed form to begin with, if you remember the folded back form that the protein was in before it opened up. And when a ligand, denoted by this ball, binds NLRP3, it's released from this auto-repressed state, it self-associates or oligomerizes, it recruits downstream signaling proteins, ASC and procaspase-1. The procaspase-1 is autocatalytically activated, resulting in activation of IL-1 beta, which is essentially an important fever-promoting cytokine. And the complex that I just showed you is what is classically called in the literature as an inflammasome, this oligomeric complex. And so NLRP3 was believed to be a cytosolic protein. And so NLRP3 was believed to be a cytosolic protein when we started this work, and so inflammasome assembly was believed to occur in the cytosol. However, what we found is that NLRP3 is cytosolic only until it's activated by a signal, here a bacterial toxin such as nigericin, but upon such activation, it associates with subcellular structures, that is the mitochondria. And we went on to find that this mitochondrial recruitment of NLRP3 was independent of all previously known adaptive proteins. Instead, it was dependent on a mitochondrial-associated protein called MAVs. And here you can see that if we silence MAVs within cells, we significantly reduce NLRP3 association with mitochondria. In cells with control siRNA, you see NLRP3 localizing to mitochondria. And so NLRP3, like I mentioned, is this protein that can be activated by a wide variety of pathogens and danger signals. So we went on and tested all of these different stimuli to see what kind of role MAVs had in regulating inflammasome responses to these stimuli. And what we found was that the role of MAVs in NLRP3 inflammasome activation is essentially non-existent in response to crystalline activators. These are mainly activators you'd come across in the context of gout, for instance, where you'd have monosodium urate crystals or calcium pyrophosphate crystals, cholesterol crystals that accumulate in atherosclerosis. However, MAVs does have a dose-dependent effect in inflammasome responses to non-crystalline activators of NLRP3, such as danger signals like ATP, which are released during mitochondrial respiration, bacterial toxins, and poly-IC, which is essentially an analog of double-stranded RNA. And we went on to find that this effect of MAVs actually was physiologically relevant. That is, in a model of acute kidney damage, MAVs knockout mice are actually as protected as known inflammasome knockout mice, denoted here by the ASC knockouts, in protecting from weight loss during kidney damage. And this protection is also associated with reduced neutrophil infiltration in the kidneys of MAVs knockout mice compared to wild-type mice, and that's essentially just quantified here. And so this is a particular instance in which activation of NLRP3 actually promotes kidney disease, in addition to its well-defined role in pathogen defense. And so, if you will, this brings us to this modified, what I like to call a mitochondrial model of inflammasome activation, whereby we think that NLRP3 is present in an auto-repressed form to begin with, that non-crystalline activators cause release of NLRP3 from this auto-repressed state, that this protein then starts self-associating. It's recruited to mitochondria via MAVs, and we think the mitochondria might perhaps act as membrane platforms for complete solid phase oligomerization of NLRP3, followed by recruitment of downstream adapter proteins. like ASC and procaspase-1, which then autocatalytically gets cleaved and activates IL-1 beta to produce active cytokine. So mitochondria, at the time that we did this work, were not implicated in NLRP3 inflammasome activation. We had some understanding that NLRP3 relocalizes to these subcellular structures. However, with this observation that MABs can be involved in inflammasome activation, we think there might be a new emerging role of mitochondrial proteins in regulating inflammatory processes. And you might have heard a lot about the recent links that have emerged between metabolism and innate immunity. And so those are some of the studies we're hoping to carry forward in this regard. And so one thing that we know is that a diverse set of activators can activate NLRP3. I mentioned that these are all the different types of activators that can do the job. We know that a common feature of NLRP3 activation is its relocalization to mitochondria. We know MABs is one such protein, and in work that my lab is currently doing, which I won't have the time to describe today, we are trying to identify in collaboration with Rob Moritz's lab here, new interacting partners which are involved in inflammasome activation, focusing our initial attention on mitochondrial proteins and trying to understand how these proteins might regulate mitochondrial functions as well as inflammasome activity. And so to kind of summarize this part, we'd like to believe that our data, along with that of others in the field, has kind of placed mitochondria as central hubs for integration of many different immune responses, whether they be responses to viruses mediated by RIG-I, or danger mediated by NLRP3, or cell death mediated by another protein called APAF1. We do know mitochondria are involved in respiration and metabolism, and recent literature has begun to establish links between mitochondrial functions and NLRP3 activation, in that we know that mitochondrial dysregulation, such as production of reactive oxygen species, even alterations in mitochondrial dynamics, things like changes in fusion and fission, can start to regulate inflammasome activation. And we think that identification of new mitochondrial partners that mediate this process might perhaps at some point yield a more unified model of how mitochondria regulate these inflammatory processes, and perhaps start offering us insights into how NLRP3 and its link with metabolism links with so many of these metabolic diseases, things like Alzheimer's disease, which we know has an underlying component of metabolic dysfunction, acute kidney disease, and the plethora of other diseases I described previously. So in the last few minutes, I will describe another story, which is essentially in line with the larger goal of the lab and towards taking a systems biology approach to understanding the functions of many different NLRs. So the NLR family, as I mentioned, it contains about 22 members in humans. We know the immunological roles or ligands for only very few members of this family. NOD1, NOD2, and NLRP3 that I just described are some of these. And what we do know from GWAS studies is that mutations and polymorphisms in these proteins are just associated with a wide variety of autoinflammatory and autoimmune diseases. Some of these are summarized here, and I won't go into any depth on this, but just to give you an idea that NLRs are associated with many diseases, but we really don't understand what signaling cascades are triggered by NLRs, and therefore we really have no understanding how dysregulation of these signaling cascades may be linked to onset of these various disease states. And so in order to start addressing this function, this question, we're basically establishing a stable expression system for NLRP3, the question being, what are the immunological functions of the undefined NLRs, and perhaps how are they regulated? And so we've established a stable expression system for NLR expression, whereby NLR expression is driven from a tetracycline-inducible promoter. And this is in a lentiviral expression system, so we can essentially take this lentiviral construct and introduce it into a cell line of our choosing. NLRs display heterogeneity in cell type expression. We think that there might be cell type specific functions of these NLRs, and so the system enables us to perhaps start querying NLR function in many different cell types. And while we're trying to do this for many different NLRs, I'll focus my attention today on describing particularly one NLR, NOD1, and another that I'll probably be describing as a control in this study, that's NLRP4. And so for both NOD1 and NLRP4, what you can see is that NLR expression is almost close to absent in absence of doxycycline, which is an analog of tetracycline that induces expression from the TRE promoter, and it can be turned on by adding dox. And once we do this, we do a series of pairwise comparisons of microarray data that is acquired from these cell lines. We need to take into account many different effects that are essentially inherent in our system. One is just the effect of adding doxycycline onto cells, the effect of leaky expression from the TRE promoter. We now know that even in the absence of any traces of tetracycline, there can be very low-grade expression from this promoter. And finally, the effect of adding doxycycline onto the cells, that is inducing NLR expression. And so quite interestingly, when we do this for NOD1, we see that NOD1 essentially just activates many, many different and diverse inflammatory pathways. And, well, I should say that these pathways are not only limited to being inflammatory, but they're associated with many different forms of cellular function, from proliferation to homeostatic signaling, to metabolic functions. The list is essentially endless. And what you'd want to note here is, even in the absence of any added doxycycline onto cells, NOD1 is able to trigger all of these different and diverse pathways. What we went on to find was that this leaky expression of NOD1 from the tetracycline promoter, just in the absence of doxycycline, accounts for about a 1.5-fold increase in the expression of NOD1. And of course, with doxycycline, you can see that that expression is elevated many-fold. We can also see this slight leaky expression of NOD1 at the protein level by Western blot at higher exposure, so many-fold higher in presence of doxycycline, but you do see very low-grade expression in the absence of doxycycline. And so this was interesting because we weren't really expecting this, essentially. But if you look into the literature, it turns out that many autoimmune and autoinflammatory diseases in recent times have been linked to mutations and polymorphisms in the non-coding or regulatory regions of the genome, which typically account for such small changes in protein expression. And so we asked the question, could that perhaps be the case? But before I move on to that, I will show you that this effect is actually specific to NOD1. It's not a general feature of NLR overexpression, because with NLRP4, you don't see any pathways being activated in absence of doxycycline, and you do see a discrete activation signature in presence of doxycycline. But NLRP4 also shows leaky expression like NOD1 does, and I've omitted the data from here in the interest of time. And so what we conclude from this is that NOD1 is perhaps activated in a switch-like manner, which we think is ligand-independent in this case, clearly, and that global upregulation of cellular pathways is specific to NOD1. Now, cellular pathways are comprised of many, many different proteins. And so what we decided to do was go back and map our microarray data onto these pathways, and then dig into these pathways to really see which genes were being differentially regulated between leaky and overexpression conditions of NOD1. And what we found was that the expression profiles are really very similar, irrespective of whether there is a 1.5-fold change in NOD1 expression or a 30, in some experiments, a 200-fold change in NOD1, increase in NOD1 expression. When we went back and looked at the genes that were either positively or negatively co-regulated with NOD1, we heard from many speakers yesterday that co-regulation might imply co-functionality, perhaps by means of genes being regulated by a common set of transcription factors. And what we found was that expression of some microRNAs was highly negatively correlated with NOD1 expression. And this was interesting because microRNAs are known to regulate gene expression in a 1.5 to 2-fold range. And this, we thought, started to offer an explanation into why such small changes in NOD1 were accounting for such large effects on cellular function. But negative regulation doesn't mean negative correlation does not mean direct regulation. And so we went back and checked whether these microRNAs are actually predicted to bind the 3' untranslated regions of NOD1. And indeed, we could see that microRNAs 15B, 106A, and 16 were predicted from 2 independent prediction databases to bind to the 3' UTR region of NOD1. And typically, this interaction takes place through what is called a seed region at the 5' end of the miRNA. And so we asked the question, do these microRNAs actually regulate NOD1 expression? And to do this, we essentially inhibited the activity of these microRNAs within cells using what are called locked nucleic acids. So what locked nucleic acids do is they bind to the 3' UTR of NOD1, that is the miRNA binding site in the 3' UTR, and they prevent the microRNAs from binding to the same site. Essentially, they inhibit the repressive effect of microRNAs on gene expression. And so you can see that we can successfully transfect these locked nucleic acids into cells. And indeed, when we inhibit activity of either miR15B, 16, or 106A, we see a subtle increase in NOD1 expression, and again, one that is distributed across the whole population. And so this is not some kind of a jackpotting effect where only very few cells in the population are accounting for the phenotype. It is a consistent increase or shift in NOD1 expression in the whole population. And that is basically just quantified here. You can see that it's about somewhere between a 30 to 50% increase in NOD1 protein. That's about a 1.3 to 1.5-fold increase. We've also observed this in ex vivo monocytes. And I'll quickly go through this to say that inhibition of miR15B and 16 similarly elevates NOD1 expression in ex vivo monocytes, again, which is quantified here. The one thing you might want to note here is that miR106A seems to have a lesser effect in ex vivo human monocytes compared to miR15B and 16. And we think this might perhaps just be cell type specific differences in regulation by microRNAs. And so finally, we went on to ask, NOD1 can be regulated by microRNAs, sure enough, but does this regulation have any role in disease? And typically, we resort to mouse models. But the one thing to keep in mind is that microRNA regulation is very different between humans and mice. And so mouse models thoroughly failed us here because the microRNAs regulating NOD1 are totally different in human cells and in mouse cells. And so we wandered into human data sets. But first, we went back to our microarray data to actually ask, where could this phenotype have any disease relevance? And what we found was that the genes most positively co-regulated with NOD1 on this graph, I previously described the microRNAs were negatively correlated. Most of the positively co-regulated genes, though, turned out to be genes that were either known tumor suppressors or proto-oncogenes. And these included genes like c-KIT, P10, CAV1, ALX1, all of which are genes that are normally involved in routine cellular function, differentiation, growth, etc. But when they're mutated or altered in function or activity, they have the potential to cause cancer. And what we found was that, sure enough, similar to what we observed on a microarray, when we validate this by qPCR, we see that regardless of a 1.5 or a 200 or 150-fold increase in NOD1, we see saturating expression of these genes, essentially saying that a subtle increase in NOD1 expression is sufficient to manifest saturating expression of these proto-oncogenes. And in recent literature, Andrea Califano has reported this recurring tumor architecture that is observed whereby proteins, what he calls master regulators, are directly responsible for regulating the transcriptional state of a tumor cell. So I'm by no means claiming that NOD1 is a master regulator, but data published by him led us to explore this possibility as to whether NOD1 might actually be totally regulating the transcriptional signature of these cells. And so to test that, we essentially CRISPR'd out only the leaky NOD1, that is the 3x flag tag NOD1 in these cells. And this is just to show you the CRISPR validation, that endogenous NOD1 expression is intact in these cells, but we're selectively ablating only the leaky NOD1. So essentially what we're doing is, in these cells, restoring the expression of NOD1 from 1.5-fold back to 1-fold. And what we see is that doing that significantly reduces expression of some of these proto-oncogenes, ALX1, c-KIT, and PTEN, some of which I showed you in the previous slide, essentially saying that the subtle increase in NOD1 was actually causal for this signature, proto-oncogene signature that we were observing. And so because we saw all these proto-oncogenes being associated with NOD1 expression, we obviously asked, is NOD1 involved in cancers? And there was a huge body of literature linking NOD1 to gastric cancer. I won't go into all the details of this, but essentially suffice to say that expression of miR-15B and 16 was shown to be previously decreased in multi-drug-resistant gastric cancer lines. We know NOD1 expression is upregulated in gastric tumors. And there's also literature showing that non-coding variants in NOD1 have increased association with gastric cancer links, so with gastric cancer risk. So taking all of this data together, we tested out this hypothesis with Ilya and Weston's help in data from the Cancer Genome Atlas. This is essentially data from a stomach adenocarcinoma data set of TCGA. And what we find is that expression of NOD1 is negatively correlated with expression of miR-15B and 16 in normal tissue. And this negative correlation is reduced in tumor tissue, which we think might perhaps be reflective of reduced regulation of NOD1 expression by these microRNAs in tumor tissue. On the other hand, expression NOD2, which is a closely related analog to NOD1, is not negatively correlated with these microRNAs. If we actually went back and looked at expression of NOD1 in gastric cancer patients, we find that there is a 1.5-fold increase in expression of NOD1. I don't know if this is just a lucky accident or this is highly penetrant, but we'd like to think the latter. But what we do find is that this increase is exactly consistent with the increase that we've observed in our experimental system and consistent with microRNA regulation of NOD1. We also tested another gene, IRF1, which was also known to be regulated by these microRNAs, and we found a similar 1.5-fold increase in expression of IRF1. And we finally asked, is expression of these microRNAs and decreased expression of microRNAs and increased expression of NOD1 associated with disease progression? And so when we look through the different stages of cancer, and what we find is that as you go through the different stages of cancer, NOD1 expression increases subtly, while the expression of these microRNAs decreases. And perhaps the data that made me the happiest was the link of this small increase in NOD1 expression with disease mortality. And so in this figure, basically, we did meta-analysis of gastric cancer data from 876 gastric cancer individuals. And what we find is that patients with high NOD1 expression have a significantly increased risk of early disease mortality in gastric cancer compared to those with low or normal NOD1 expression. And so I think it's time for me to wrap up. And I will say that I've omitted much of the data here, but our working model is essentially this. We believe that although NOD1 has evolved to be a sensor for bacterial pathogens, and its most well-known function is essentially to activate NF-kappa B, resulting in activation of pro-inflammatory genes within the cell, under normal conditions, this does not happen because NOD1 expression is kept in check by microRNAs that hold this whole signaling cascade in check. However, if this regulation is lost, perhaps due to mutations in microRNAs or mutations in the 3' UTR of NOD1, or one of many other factors in cancer that might affect either NOD1 or microRNAs, then we get an increased flux through this NOD1 signaling pathway, which is associated with a small increase in NOD1 expression. essentially resulting in what we observe is an altered cellular transcriptome and increased levels of proto-oncogenes with the potential to cause cancer. And we think part of this might just be because you get increased expression of pro-inflammatory genes downstream of NOD1, and a persistent inflammatory microenvironment has been linked with onset of cancer. And so with this, I will thank the people who did the work. The NOD1 data that I showed you, the work that I showed you was done by Leah, Ajay, Carolyn, and Cliff, so pretty much a joint lab effort, if you will. Henry and Owen are working on the interaction of NLRP3 with mitochondria that I didn't update you on much today. I'd like to thank Ilya's lab for initial help with analysis of gastric cancer data. I think I mentioned other people along the way, and of course, I'd like to thank Ron Germain, in whose lab some of these initial systems were set up. And with that, I will take any questions, if we have time, but we probably don't.
So if there are any questions, perhaps I could ask one.
They're speechless.
My question is about NLRs localizing through MAVs to mitochondria. And you speculate that it might have to do with ROS signaling. However, lots of other organelles produce ROS, peroxisomes, ER, and so on. And so I'm wondering whether there's localization to other organelles for NLRs, and whether you've investigated that with your 22 NLRs.
Right, so the answer to the second question is no. Having said that, we have no data to show that localization of NLRP3 to the mitochondria is mediated via ROS, essentially. What we think is, the way we think of the model is essentially that localization to mitochondria is a very early process, essentially. We haven't measured ROS, and so I won't be able to comment on that. But we think that the mitochondrial membrane perhaps then nucleates full oligomerization of NLRP3. And at that point, you might have production of ROS, but again, we haven't looked at that. With regard to relocalizing NLRP3 onto a different organ, I think that's a really interesting thing to do. MAVs has been known, is known to be present on peroxisomes as well. So one of the things we're trying to do is create cells that have MAVs only on mitochondria or only on peroxisomes, and then try to see whether we get a functional inflammasome complex even from peroxisomes. And in fact, if that is true, that it is simply the requirement for a membrane platform, or perhaps requirement for cellular signals that are common to some of these organelles, then we would hypothesize we'll see functional inflammasome activation from peroxisomes as well.
Terrific, thank you. So with that, is there a question there?
You're actually referring to most of my question, that last part of your answer there, of MAVs being located to also peroxisomes and to the mitochondrial ER MAM membrane. And do you think that there are different functions of NLRs going to the MAVs that is associated to those different organelles?
I don't know. It is something that we're trying to test. And so I, of course, answered John's question, and this is really the outstanding question. What do mitochondria actually do in this case? And perhaps for other NLRs that relocalize to different organelles, what do those organelles do? But we're just starting to get into that. The question really is this simply a requirement for a membrane platform and only for MAVs, or is this an active modulation of inflammasome function by organelle-associated proteins? And so what we're hoping is that redirecting inflammasome activation to peroxisomes will start yielding answers to some of these questions. If it is, in fact, an active regulation, then we would hypothesize we'd see some differences in inflammasome function from these different organelles.
Thank you, Neha. So thank you very much, Neha.